isotope was incorporated or not easily passing into the newly synthesized DNA strand. Okay. This heavy DNA molecule could be disintegrated from the normal DNA by centrifugation method. Okay, this centrifugation was done in a cesium chloride medium where density gradient is present. Okay, this density gradient is between, note that 15th nitrogen is not a radioactive isotope. Okay, it can separate it from the 14th nitrogen isotope. These isotopes helps to detect the genetic material. Now finally, this is an experimental set of Meselson and Stahl. This is the first step. Gravitational force was applied and you can see 15th nitrogen DNA was 20 minutes incorporated. Okay, and you can see here NDNA generation 1. This is the generation 1 nitrogenous DNA. Why? Because isotopes were incorporated into the DNA, and this is generation 2 genetic material with the nitrogen DNA. Okay, isotope DNA. This isotope labels the specific sequence in the DNA. That's why Meselson and Stahl used this nitrogen isotopes. Now then they transformed the cells into medium with a normal 14th nitrogen NH4CL ammonium chloride and took samples at various definite time intervals, okay, when the cells are multiplied, and extracted that DNA was remained as a double stranded helical. The various samples, if you carefully observe, the various samples were separated independently on cesium chloride gradient to measure the density of DNA. Thus, DNA that was extracted from the culture 1 generation after the transfer from 15th nitrogen isotope to 14th nitrogen isotope medium, that is after 20 minutes, okay, after 20 minutes, E. coli divides, okay, in 20 minutes, had a hybrid intermediate density. DNA extracted here and from the culture after another generation, that is after 40 minutes of time, okay, second generation was obtained. In the diagram I have shown you what is the second generation DNA segments. If E. coli was allowed to grow for 80 minutes, how many minutes? 80 minutes. Then what would be the proportion of light and hybrid densities? Let me ask you this question. Very simple experiment involving using radioactive isotopes, nitrogen, okay, involving radioactive thymine or radioactive thymidine, which can detect the distribution of newly synthesized DNA in the chromosome, which was performed on plant-based beans called fabia beans or Vicia faba by Taylor and his colleagues in 1958. The experiment proved that DNA in chromosomes also replicate semi-conservatively.